let's go to the next slide. So let's look at that muscle stuff a little bit more in detail. Muscle cells, or fibers we call them also. So when we call fibers, we mean cells, or it's the same, aka. They're unique in that their main function is to contract. So that's all they do. And so they have, inside, they have specific structures, specific organelles that are specific to them. So for once what we have is we have a membrane that is reactive. So the cell membrane is called a sarcolemma because it can transmit an electrical impulse, which is necessary because when we want to contract the muscle, a skeletal muscle particularly, we have, well, all of them, but you know, we start talking more and more about the skeletal muscle here. We want to have that contract, we need to stimulate that. So it's excitable, but it needs to be tickled, it needs to be irritated. So the sarcolemma can now, is reactive, that it actually can carry a nerve impulse into the cells. And so that's going to be interesting. Then we have myofibrils, and we talked about these thread-like, look, this is actually a very nice slide. We talked about these thread-like long parallel arranged fibers that shorten, that contract, that go past one another, or one pull on the other. That's inside here, and one of those units that has a properly arranged threads inside of it is known as a myofibril. And the myofibril goes the whole length of the muscle, and they're parallel arranged. So this is a whole muscle cell, and on the inside we have again looking things like another muscle cell looking thing. So it's like from bigger to smaller. It just gets smaller, but it kind of looks very similar. And so that's and then we have a third thing, and we're going to start talking about the third thing in a minute here. That's that blue stuff here. But first, let's look a little bit more at that sarcolemma, because that sarcolemma actually projects inside the cell, surrounds each myofibril. So when a nerve impulse comes in, it can go and tickle that myofibril on the inside and bring that irritation deep, deep inside. So the muscle all of a sudden can contract all at once. And so that is those deep, deep penetrations are known as the T-tubules. And they envelop the myofibril. And then what we have is we have this other stuff, we have this blue stuff that is in between these penetrating fibers that bring the nerve impulse into the fibers, into the cells. And that blue stuff is actually specialized endoplasmic reticulum that acts like calcium bags. And so it's known as sarcoplasmic reticulum. And in this system, the T and the L system, don't really ask me where that came from, but it's known as the L part of the system. And so this nerve comes in now, and what it does, it stimulates the SR to release the calcium deep into the muscle cell, deep into the myofibril, and talks to every one of those single threads. And what it does there on the threads, it makes sure that these threads can connect to one another. Otherwise, they're just threads, they just hang there. But when the calcium is present, they are able to connect to one another, and one can attach to the other one and pull on it. So that's kind of really where we go with that. A myofibril is so the shortening, contracting organelle in a muscle cell. So it's, when we go deeper on the inside, it is composed of these two thread-like proteins. They're threads. And one is a thick one, that's called a myosin. And the other one is a thin one, and that's called the actin. And they're myofilaments, and that means they're protein strands. And they are arranged parallel along the whole length of the muscle. One of them is not the whole length of the muscle, so one of them is very short actually. And when we look at when we go back here for a second and we look at this slide here, we see these up and down lines, these white up and down lines that are perpendicular to the parallelness of those little threads, these actin and myosin fibrils. when we look at those up and down fibers are actually the anchoring places for the small, thin filament, the actin filament, these Z discs, they call them. And so we can see that on the microscope. And so when we want to look at small subunits that we can study of how does a muscle contract, we chop that myofibril in small little pieces that go from a Z disc to another attachment site of the actin on the other side, another Z disc. And so we call that a sarcomere. I'm sorry, there's all these names, you know. I remember that because it's merely a muscle. It's so small, so that's a sarcomere. But, you know, whatever works for you. And so these actin fibers, these little threads of protein are anchored on these C discs and penetrate inward towards the middle of the sarcomere. And then from the middle, where there's actually a midline, there's also sort of an anchoring point for the myosin threads, the thick ones, and they penetrate and go and go past the actin ones on both sides. The thin filament is basically is a thread which actually has another thread on top of it, and then some small little stuff. What happens is this here is the thread that this bigger thing down here, this myosin one, needs to attach. And the myosin has a head, and the head is what attaches to the actin. And then it has a tail, and the tail is what's anchoring to the midline. And so the head needs to attach to the actin, and so the actin actually has these sites, they're called active sites, where the head can attach. But when that little thread, called Tropo Myosin, in other words, that we have, when that little thread is overlaying the active site, that head cannot attach to it. And so that's the state when the muscle is relaxed. And when the muscle needs to contract, that active site needs to be open up so the head can attach to it and actually do the work. And the molecule that will do that movement of the tropomyosin and remove it and free up the active site, that is calcium. So that's the role of calcium that we'll get into a minute again. But so let's go a little deeper and let's have that muscle shorten. And so the actin and the myosin slide past one another, shortening the sarcomere while preserving the length of the actin and the myosin. Okay. So what actually happens is that the myosin head is attached to the actin active site, and then this part of the head is sort of in a position that is activated. See here the ADP, not ATP, ADP and the phosphate. So it's detached to phosphate. That means the work is already done in terms of the energy work. So in this state, it attaches to the active site, and then from there it moves into the lower energy state and does the work, like the spring gets released, so to speak, and the ADP and the phosphate fall off, and we bring in and it detaches then from the active site, while an ATP comes in, a high energy molecule, and works on it and puts it back into this active state. And then see that's hydrolyzed, the ATP is hydrolyzed again to ADP and phosphate. So it's removed the energy bond. And so transferred the energy to the myosin head. And then in this position again, it can attach to another active site on the actin because it's freed and does the work again. And so that is known as the sliding filament theory. That's how we move. The revolving of that process brings the myosin closer to the Z lines of the attachment sites of the thin filaments of the actin. And here is the reason why we have the calcium again. In order for the myosin heads to not always attach to the actin protein thread, so-called active sites on the actin are covered by tiny protein threads. Those are the tropomyosins. So these little threads here, tropomyosins. While the muscle is inactive. And calcium is needed to remove the tropomyosin when muscle gets stimulated by a nerve impulse. And so that's what happens here. We can see a cross-section where the calcium molecules come in and work on this other little small complex, the yellow one, that's the troponin, and that pulls the tropomyosin away from the active site so the head can attach to it. Oh boy. Yeah, this is complicated. So let's do the whole thing in a whole picture, big picture piece, okay? So here, number one, we get a nerve, that's a nerve, that brings down, and what happens when a nerve actually goes to and attaches to something, a chemical gets released. And so a nerve impulse is electrical, but the thing that makes the change on the next structure is actually a chemical. So the chemical here that gets released is ACH, that's acetylcholine. Don't worry about that name. It's a neurotransmitter. So it's that chemical that brings over the information that says like, hey, we got to do something, we got to contract. And so the action potential, that impulse, that's an impulse, gets generated and propagated along the sarcolemma, the cell membrane of a muscle, and into the T-tubule, deep into the penetrating around those myofibrils. And from there, it triggers the calcium release from what they call the terminal cisternae of the sarcoplasmic reticulum. So the calcium goes into, the ion goes into the area where we have then, into the myofibril, that organelle where we have the actin and the myosin. And so it binds into the troponin. The troponin changes shape, removing the blocking action of the tropomyosin, which is that small little thread gets removed. And then the actin active site is exposed. And so what happens there, the myosin cross bridge, the head, can attach to the actin and pull on it as it detaches and gets activated again. And so that pulls the actin filaments towards the center of the sarcomere, that little small studyable structure. And then release of energy by ATP hydrolysis powers this cycling process. So yes, ATP powers that part. ATP powers the myosin. The calcium allows it to happen. And then when the calcium is removed by active transport back into the sarcoplasmic reticulum, after the action potential is finished, after the nerves start firing, guess what? The tropomyosin blocks the active site again, and the actin is not the myosin is not able to attach to it, and so the contraction ends and the muscle fibers relax. Well, there you have it. I think that's a pretty good slide. So I think we should make